Hello, everyone. Welcome to today's webinar. I'm Gina Vitale, Assistant Editor at CNEN, and I will be moderating today's event. This webinar is titled Reverse Phase Liquid Chromatography, Fundamentals and Strategies for Faster Method Development, and is being sponsored by Advanced Materials Technology. CNEN works with sponsors to identify topics of interest and value to CNEN's audience, and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN online after the live webcast. The presentation today is being sponsored by Advanced Materials Technology. Advanced Materials Technology Company is an innovative product development and manufacturing company located in Wilmington, Delaware, that is dedicated to researching and manufacturing high-quality enabling materials for scientists around the world. They primarily support market segments such as pharmaceutical research, biopharma, medical research, and toxicology. The team at AMT is committed to and best known for manufacturing innovative products and being the leader in on-time delivery of the highest quality separation products needed in the testing and development of pharmaceuticals and biopharmaceuticals. During the presentation, we will hear from Dr. Stephanie Schuster, who is a Senior Technical Support Scientist at AMT. Dr. Schuster joined Advanced Materials Technology in 2009 as a research scientist, providing contributions to the commercial development of products designed specifically for the separation of peptides and proteins. Most recently, as part of the technical support team at AMT, she has been providing customers support in optimizing and troubleshooting their applications. I will now hand it over to Dr. Schuster.
Thank you, Gina, for the introduction, and thanks to everybody for joining us today. So I'd like to talk about reverse phase liquid chromatography method development. And here's a brief outline of what we'll cover today. In terms of the fundamental metrics, we'll review efficiency, selectivity, and retention as it applies to the resolution equation. And then we'll go into tools to help select columns, including the hydrophobic subtraction model and the Tanaka test. And then sprinkled in throughout will be real-world applications and method development scenarios. And then we'll wrap up with a summary. So what are the 3 steps for basic method development? Well, first you need to know what are your target analytes and what kinds of information do you have about them. So for instance, do you know the pKa, the polarity, the solubility of these compounds? That's really the first step. Second, then, you would ask yourself, well, what columns should be screened? And in order to answer that, you need to know something about the selectivity of the stationary phases of the columns that you'll be using. And to help answer that question, you can either use the hydrophobic subtraction model or Tanaka testing, or even a combination of the two. And then finally, you need to decide what mobile phase conditions should be used in terms of should you use a gradient or isocratic conditions? Is it better to use methanol or acetonitrile? And then finally, look at temperature and also the effects of pH. So, of course, the goal is to resolve all the peaks in the shortest time possible. So now I'd like to sprinkle in a couple of polling questions here. For the audience, what is your biggest challenge in method development? Lack of chromatographic resolution, the runtime or throughput, chromatographic reproducibility, column lifetime, or selecting columns? And you can choose more than one. I'll just give you a few more seconds to submit your answers. Okay. So it looks like For our audience, lack of chromatographic resolution is the most popular challenge here, followed by selecting columns. And I think that probably applies to most people starting out their method development. You know, the main goal is to resolve all those peaks. So hopefully, by what we're going to tell you in the webinar, you'll have some guidance on what to do in making those column choices and getting the resolution that you need. So thinking about step 1, these are some example target analytes. And sometimes you know what analytes you're looking at, and other times you don't. I just have included a couple of different examples here. So for instance, there's fentanyl, pain medication, acetaminophen or Tylenol, hydrocortisone, which is a steroid, warfarin, an anticoagulant. So you notice these compounds have different functional groups. They'll have different pKa's, different solubilities, and they lend themselves to be analyzed basically with different conditions. So if you know the target analytes that you're trying to separate, that's really a good place to start for your method development. So I have another polling question here. How much information do you typically have about your compounds? The log P, pKa, the structure of the compound or functional groups, maybe some of the above, or I wish I had this information. So I'll give you some time to submit your answers. Okay, a few more seconds to submit your answers. And let's see. So looks like most people have at least the structure of their compound in terms of functional groups. They might have some of the information. And then some analysts are wishing they had that information. Right? So this is really one of the challenges of method development is not really knowing what exactly is in your mix of analytes to be separated. So this leads us to step 2, which is the column choice, and in particular, the selectivity of those columns. So just to take a step back, I'd like to just refresh everyone on what is selectivity or alpha. This is also known as the separation factor. And it's defined as the space between peaks in an HPLC separation. It's the ratio of retention factors, or K, values between neighboring peaks and is always 1 or greater. So it's defined as a K2 divided by K1. And in this example here, I'm showing a K value of 9 divided by 4.26 gives a selectivity of 2.11. And, this is an exaggerated example of really great selectivity, but sometimes you're not quite so lucky. So why are we so interested in selectivity? Well, that's because it really plays a key role in the resolution equation. And here I have the resolution equation with its 3 different terms. Column efficiency term listed in green font, the selectivity term listed in red font, and the retention term listed in blue font. So we're sort of taking for granted that we have adequate retention, so I'm not going to spend much time talking about retention in this particular webinar, but I am going to talk about, in particular, column efficiency. And what is column efficiency impacted by? So you have particle size, which affects efficiency, the column length, of course, dispersion, and sometimes even the particle pore size can impact the efficiency that you see for your separations. So this is where I'd like to tie in the advantage of using superficially porous particles. So just as a reminder, what we have is a solid silica core with a porous silica shell. And you can see the SEM on the bottom left-hand side of the slide here. And what we're showing is the 90 angstrom pore size in 2.7 micron total particle size. And the advantages of that solid core's short diffusion path, here we're showing on the bottom right, the diffusion path is a half micron thick shell. What that enables is high efficiency because the analytes don't have to travel as far into and out of the porous shell where the bonded phase is. And what that enables is to run faster flow rates, so that gives you speed, and that will help you more quickly develop your methods. And then finally, you have a relative lower back pressure compared to sub 2 micron particle columns, because the particle size of the superficially porous particles is bigger compared to the sub 2 micron. So if you're running at lower back pressures, that's less wear and tear on your instrument in the long run. So now, going back to the selectivity term of the resolution equation, the reason that we're very interested in selectivity is because resolution is almost linear with selectivity. So the better your selectivity is, the better your resolution is going to be. And what parameters influence selectivity? So you have first your column type, and what I have starred here, those are more effective at influencing selectivity. First, the mobile phase will influence your selectivity. The pH, especially, in particular for ionizable compounds. Temperature. Ion pair concentration. The percent B organic solvent or gradient slope. And then finally, buffer concentration. So you have all of these different parameters influencing selectivity, and I'm going to be really focusing in on the column type or stationary phase for this discussion. So how do you navigate the column catalog past C18? So here you are trying to develop a method. You open up a column catalog, and you know most people are familiar with C18 phase, but what about all those other phases? How do you decide which one to pick? So this is the part where you can use the hydrophobic subtraction model or the Tanaka Testing characterization in order to put these different bonded phases on an even playing field and compare their selectivity characteristics and what really gives them their retention behavior. So next I'm going to talk about the available tools to help select columns, including the hydrophobic subtraction model and Tanaka testing. And I have one last poll question here. I'd like to know, do you use any of the following when starting method development? Modeling / method development software, visual characterization techniques, for instance, Tanaka plots, hydrophobic subtraction model, or HSM, in particular using the database at hplccolumns.org, combination of the above, or none of the above. And I'll give you a few more minutes to submit your answers. Or not minutes, a few seconds. So we have none of the above. Seems to be the majority of attendees today. So that's pretty interesting. So that leads me to wonder, how, what are most people doing to start out their method development? Well, there are multiple ways to start method development, and no one way will really fit each and every scenario. So we'll talk about that more as we go along. So the hydrophobic subtraction model. What I have here is the equation, and what we're doing is taking the log of retention of a particular solute retention factor divided by the retention factor of ethylbenzene. That's the EB on the lower K value. And what that is related to are the lowercase coefficients, which actually relate to the different solutes that are used in the model. And then what happens is multiple linear regression is used to calculate the capital letters. So for instance, H measures the hydrophobicity of the phase. S star measures the resistance of the stationary phase to penetration by a solute molecule. A measures the hydrogen bond acidity of the phase. B measures the hydrogen bond basicity of the phase. And C measures the interaction of the phase with ionized solute molecules and is measured at both pH 2.8 and 7. So what you can do is go to hplccolumns.org, which is a database of over 700 columns worth of data. And what the idea is, these tests have already been done and the columns have already been characterized. So what you can use it for is to determine bonded phases that are similar to each other or actually different from each other. So if you know you have a C18 phase, for instance, and you want to know what is the most different or orthogonal in terms of its selectivity, you can go to the website and compare the columns to each other. And the way that this is done, practically speaking, you would select your column that you want to compare to. So in this case, I'm comparing to the Halo C18. So that's why the F sub S value here is 0. And the F sub S is actually how different the coefficients are from one phase to another with a weighting factor. So in terms of column selectivity, if the F sub S value is greater than 12, those columns are considered orthogonal, or they have a very high chance of giving different separations. Whereas if the F sub S value is less than 12, then you would have similar selectivity, and you wouldn't expect very big changes in terms of the separation. So if I was to pick something that was different from C18, I could come halfway down the list here and compare to a halophenylhexyl, which has the F sub S value of 17.35. And then also in that same column set, I might choose the halo RP-amide, which has a F sub S value of 52.83. So I have an example of how the selectivity of a separation changes across these 3 different phases. So utilizing the HSM for method development, what it's really doing is helping you select columns that have different selectivities from each other. So here I'm showing the separation of paracetamol or acetaminophen and its associated impurity compounds. The top trace in blue is halo C18, the middle trace in red is using the halo RP-amide, and the bottom trace in black is using the halophenylhexyl. So first let's look at the difference in retention and resolution between compounds A and B on the halo C18 column compared to the halophenylhexyl column. So you can see the B compound is more retained on the halo C18. And what do those compounds actually look like? So here I have the structures of all those compounds, and we were focusing in on compound A and compound B. So A, you can see, has it's less hydrophobic than the B compound and for hydroxyphenylpropanamide. And this is the explanation for why the halo C18 column is is more retentive for this more hydrophobic compound compared to the halophenylhexyl. And I have another example comparing the halo C18 to the halo RP-amide for compounds F and M. You can see they're less retained on the halo C18 column, and then we're seeing increased retention on the halo RP-amide column. So let's take a look at those structures. Compound F is 4-nitrophenol, and compound M is 4,4' azine dilyl diphenyl. So the explanation for why are we getting more retention on the halo RP-amide phase is because of the presence of these hydroxy groups, which we normally see increased retention on RP-amide phase with presence of hydroxy groups. So that's what's impacting the separation in this case for the halo RP-amide compared to the halo C18. So now I'd like to switch gears and talk about Tanaka testing. So this was a group of tests developed by Tanaka in 1989, and the columns are subjected to 6 different isocratic tests in order to characterize their selectivity. So similar to the hydrophobic subtraction model, we're trying to characterize the column's hydrophobicity, the shape or steric selectivity, hydrogen bonding, ion exchange at both pH lower than 7 and pH higher than 7, those environments. And then the results are typically displayed in a radar plot to visually compare the phases. So much like the data is available in a database, this Tanaka testing data is also available via the ACD Labs website, and you're able to compare columns directly there as well. And we like to say this is a way to visually compare columns because the data, like I said, is normally plotted in a radar plot. So if you direct your attention to the bottom right-hand corner, you can see I have a radar plot with 5 of the different tests plotted here. And I'll talk about the reason why there's 5 in a minute. So normally the 6 tests that would be run for Tanaka include the A and the B test, which are actually the same thing, which is the retention factor of amyl benzene, or actually it's also known as pentyl benzene. So for the Euerby-modified Tanaka test, in order to simplify that, they remove one of the axes and they just look at the retention factor of pentyl benzene one time. So that's how you go from the 6 isocratic test to 5. So in terms of hydrophobicity, what we're looking at is how retentive is the column for pentyl benzene. For the shape or steric selectivity test for the C, we're looking at the retention factor of triphenylene. For hydrogen bonding capacity, this is a measure of the amount of silanols, end capping, and surface coverage, and that's given by the retention factor of caffeine. For the ion exchange capacity, greater than pH 7, this is a measure of the amount of silanols and ion exchange sites, so that's given by the retention factor of benzylamine. And then finally, test F is the ion exchange capacity at pH lower than 3. This is a measure of the amount of silanols and ion exchange sites at pH 3 and whether there had been any silica pretreatment. And that's measured by the retention factor of pentyl benzene as well. So in terms of visualizing selectivity differences, the 5 tests that I've just described, those results are plotted starting with hydrophobicity at the top of the radar plot, and then working clockwise around so to shape selectivity, hydrogen bonding capacity, the total ion exchange, and then the acidic ion exchange. So here are the 10 bonded phases that are available on halo particles for small molecule separations. And you can clearly see that the different bonded phases have different characteristics, which would then help you in choosing which column to use for your particular analysis. So I have a couple of examples here. First, I wanted to talk about hydrophobicity. So in terms of one of the most hydrophobic phases available on the halo particles is our C18. So what's done is the results are normalized to the phase that has the greatest hydrophobicity in order to have everything on the same scale. So you can see that the most dominant characteristic of C18 is its hydrophobicity. It doesn't have much shape selectivity, and it doesn't have much hydrogen bonding capacity. And the same thing for total ion exchange and acidic ion exchange. So the take-home message here is that depending on what kind of separation you're trying to go for, a C18 phase would not be very shape selective or able to really participate in hydrogen bonding capacity. And what that means is there aren't very many free silanols on the surface of this phase. So now, if you did want a shape selective phase, we would recommend our halo PAH, which is specifically designed to analyze polyaromatic hydrocarbon compounds. So on the top right corner, you can see the Tanaka plot where the C18 traces in blue and the PAH traces in green. And you can see in terms of a shape selective phase, the PAH is our most shape selective phase. And this is really key in order to separate the polyaromatic hydrocarbons, which have pretty similar structure. So I've circled a couple of pairs where they were co-eluted on the C18 column in the top blue trace. So in particular, compounds 5 and 6, that's ace-naphthene and fluorine, those are baseline resolved on PAH phase here on the bottom green trace. Compounds 11 and 12, you're seeing an elution order reversal. Compound 11 is benzo-A-anthracene, and 12 is chrysene. And you can see you're getting baseline resolution on the PAH phase compared to the C18. And then finally, compounds 17 and 18 are co-eluted on the C18 in the top blue trace, but they're resolved in the bottom green trace. And those compounds are benzo-GHI-pyrroline and indeno-1,2,3-CD-pyrene. The other interesting thing here is, in terms of hydrophobicity, C18 is a bit more hydrophobic for the first portion of the separation up to compound 10, pyrene. And then after pyrene, you can see that the PAH phase is more retentive for those more hydrophobic compounds. I have another example of shape selectivity using tocopherols or vitamin E. So first direct your attention to the top right corner where I have the Tanaka plot. Again, the C18 Tanaka traces in blue, and for PFP we have it listed in brown. So you can see that in terms of shape selectivity, the PFP phase has a higher value compared to the C18. And then in terms of hydrophobicity, the C18 column is, of course, more hydrophobic than the PFP. So we're looking at tocopherols, like I said, and in terms of the R1 and R2 groups, you have methyl groups at R1 and R2 for alpha tocopherol, a methyl group at R1 for beta tocopherol, a methyl group at R2 for gamma tocopherol, and no methyl groups for the delta. So in terms of the separation on the C18 column, you can see that the gamma and beta versions, which it just depends where there's a methyl group, those aren't resolved on the halo C18, whereas they are resolved on the halo PFP column. And the other interesting thing about this particular comparison is the difference in retention time. So you can see the separation is complete about 8 times faster on the halo PFP phase, which is much less hydrophobic than the halo C18. So in this case, retention really doesn't dictate whether you will be able to separate the compounds of interest or not. So sometimes I feel like that might be a little bit counterintuitive, but it's really the dominant part of interactions here is that shape selectivity for this particular analysis. And then next I'd like to show a hydrogen bonding capacity example between a halo C18 column and a halo biphenyl column. So again, the top right corner is showing us the Tanaka plots, and we can see the C18 trace in blue and the biphenyl trace in lavender, and you can see that there's more hydrogen bonding capacity for the biphenyl phase, which would mean there would be more available silanols to help with the separation. So here we're separating a mix of 12 different beta blockers, and I've pointed out the structure of propranolol, compound 10. You can see that's more retained on the halo biphenyl column compared to the halo C18, and you can see that propranolol, has some polar moieties here, so that's part of the reason why it's more retained on the biphenyl column, because it's able to interact with those silanols. And the same thing for the last compound, compound 12. It has more retained on the halo biphenyl phase compared to the halo C18. And those are all the examples that I had for selecting columns based on the Tanaka plots. But if anyone has any questions about that, I'd be happy to answer them at the end of the webinar. So now, step 3, in terms of mobile phase conditions, now that you would be able to select a set of columns that you could use for method screening, the next part would really be deciding which mobile phase conditions to use. So in this example, I'm looking at a set of steroids run on the halo C30 column, and we're not getting resolution for compounds 3 and 4, prednisolone and hydrocortisone, using acetonitrile as mobile phase B. If we switch to methanol as mobile phase B, you can see now we are able to resolve compounds 1 and 2, and particularly 3 and 4, but we're not quite getting baseline resolution because here the resolution is 1.2. So if we switch to a 50-50 mix of acetonitrile and methanol, you can see we're able to increase the resolution to 1.7 for compounds 3 and 4, the prednisolone and the hydrocortisone, and we have a good compromise in terms of the total retention for this particular set of analytes. So what I'd like to point out from this example is you may not You may not get the selectivity that you want with 100% acetonitrile or 100% methanol, but sometimes the combination of acetonitrile and methanol is really something that is a good idea to try in order to get the resolution that you need. And then, of course, pH has a big effect for ionizable compounds. So depending on the nature of the analytes, changing the pH of the mobile phase can have a large impact on the selectivity of the separation. So the pKa of 3-nitrobenzoic acid is 3.46. And at pH 7, the top chromatogram, the 3-nitrobenzoic acid is in its anionic or charged form, which explains its lower retention compared to when the compound is neutral at pH 2, bottom chromatogram in blue. So you have to be careful to use a pH that is 2 units away from the pKa of the analytes in the separation, or else the compound could possibly exist in 2 different forms, which could show itself as split peaks or a kind of odd peak shape. And then finally, of course, temperature does play a role in terms of the separations and how well the compounds will be resolved. So in this example, I'm looking at trans vitamin K1, which is peak 1, and cis vitamin K1, which is peak 2. So what we're showing here, in particular for the Halo C30 column, is as we decrease the temperature, we actually are able to increase the resolution. So for the A condition, we're running at 35 degrees C, and the resolution is 1.53. If we decrease the temperature by 5-degree increments, you can see that we start to get baseline resolution just by running at 30 degrees. And we're able to go all the way up to condition E, which is 15 degrees C with a resolution of 3.03. So of course, you would need an oven that can cool to below ambient temperatures in order to do this separation. But in terms of different isomers, sometimes running at lower temperatures is an advantage and will help you get the resolution that you need for your separation. So then in terms of a summary and conclusions, you definitely want to use the knowledge of target compounds if it's available to you for your method development. Selection of an appropriate column phase or a set of phases is enabled by using the hydrophobic subtraction model and Tanaka testing. Once those stationary phases are identified, you want to screen both the organic solvent and pH, then vary temperature and gradient time. And we recommend using a commercially available method optimization software package in order to develop robust methods. So you're not operating on the hairy edge of a good separation. And finally, it's good practice to screen multiple phases to ensure the best possible separation when screening an unknown mix of compounds. And I have a few acknowledgements. I'd like to thank the team at Advanced Materials Technology, especially Stephanie Rosenberg, Connor McHale, Andrew Harron, and Ariane Sullivan. So with that, I'd like to turn it back to Gina and thank everyone for their attention today. I'd be happy to answer any questions you might
have. Okay, we're going to start with this question. This says, interesting to see that the AQ C18 is most orthogonal to normal C18. Can you say something about that?
Oh, sure. So, actually, let me go back to my hydrophobic subtraction model data. So the Halo AQ-C18 is actually very similar to the Halo C18. One of the things is these hydrophobic subtraction model tests are run using acetonitrile, and what we found is we actually get more selectivity differences between C18 and AQ-C18 using methanol mobile phases. So I think just to clear any confusion up there, we're actually, maybe you were thinking about the Tanaka. Let me push that. But in terms of the hydrophobic subtraction model, the C18 and AQ-C18 are really pretty similar. And I don't know, in terms of really pulling out the AQ-C18 versus C18 here, you can see there's more total ion exchange for the AQ-C18 versus our standard C18. But technically, I wouldn't say it's the most orthogonal.
Okay, we can move on to another question. This says, would you have recommendations for column stationary phases to try for separations of excipients, surfactants?
That's a great question. So surfactants are a bit of a tricky analyte because they're normally Twitter ions. So you would have to find a phase that could really, give you some good retention of those particular compounds. I think I might try maybe the RPLC phase for that, but I could always do a bit of background research and get back to you on that one.
Okay. This question says, what detectors did you use in the method development, UV-Vis or mass spec?
Oh, that's a great question. So for the examples that I showed today, we used all UV-Vis, but we have in the past also used mass spec.
How can we decide which column is best for a mix of unknown analytes?
Okay, well, when you have a mix of unknowns, it's best to screen several different stationary phases, which would give you different selectivity. So like I had mentioned earlier, I would recommend screening, of course, C18, since everyone likes to start out with C18, RP-amide, and then phenylhexyl.
Okay, this question says, thank you for the helpful talk. How can we know worth trying to mix organic solvents like methanol and acetonitrile as mobile phase B for improving selectivity?
Oh, that's a good question. So you really don't, there's no way to predict that unless you actually try it. There are some method optimization packages where you could possibly model the mixing for you. But if you don't have access to that, I would just say, if you're noticing that, acetonitrile isn't giving you the selectivity that you need, and methanol isn't doing the job either, then I would say it would be a good idea to try mixing them.
Would you have any suggestions for peptide analysis with nano-LC? Reproducibility is often an issue also.
Sure. So, in terms of nano LC, I would say one of the really key things for running nano is to keep that instrument running constantly, 24/7, in order to not have any bubbles in the system, and then um, maybe for peptides, we do have particular other bonded phases and different pore sizes that we would recommend. So I think in order to really get reproducible data, I would recommend, if you're not using some kind of oven or temperature control, that can impact the reproducibility. And then also depends on the acid modifier that you're using. So if you're going into a mass spec, I would recommend difluoroacetic acid, because that gives you a good compromise between the ionization efficiency that you get and nice symmetrical peak shape. If you're just using formic acid at the moment, sometimes you can see different results just because, the formic acid will evaporate over time from the mobile phases. So if you're not using mass spec detection, if you're just using UV, then I would recommend using trifluoroacetic acid for some good reproducible results.
Okay. Can you apply the Tanaka modeling and hydrophobic subtraction models to peptides and oligonucleotides?
Oh, that's a great question. So we haven't really tried to do that, but I think it is possible. Of course, those particular phases would have to be screened by the model, and then you'd be able to better characterize the contributions that are contributing to retention.
Okay. Do you have any recommendations for mobile phase modifiers to assist peak shape?
Sure. So if you're using UV detection, like I mentioned earlier, I'd recommend using trifluoroacetic acid, or TFA, for its ion pairing capability. And of course, this is running at low pH. If you're using mass spec detection, then I would suggest difluoroacetic acid or a mix of formic acid with ammonium formate for good peak shape. And it's also possible to use mixes of TFA and formic acid for a compromise with good peak shape and ionization efficiency.
Any comment on acidifying mobile phase with formic acid relative to column type / performance?
Oh, sure. So it really depends. If you have, say, a base deactivated phase, then formic acid will give you fairly good peak shape under those conditions. But if the column has free silanols and you're trying to use formic acid, then you're probably not going to see very good peak shape of your analytes.
Okay. For metabolite analysis, especially those complex mixtures of various polarities, do you have a recommendation for a stationary phase or phases that would be best to initially screen?
Yes, I would. So when you have those really polar compounds, sometimes you need to start at gradient conditions that have no organic or very low organic. And in those cases, you need a phase that is 100% aqueous compatible. So I would recommend RP-amide phase, for instance, because that's 100% aqueous compatible. Of course, our AQ-C18 is 100% aqueous compatible. The biphenyl and phenylhexyl phases are also 100% aqueous compatible. So I would start out with any of those four. I would not recommend C18 or C8 because those phases are very hydrophobic and they can't be used under 100% aqueous conditions.
How does the isocratic and gradient mobile phase flow help in attaining good peak separation?
I'm not exactly sure what is meant by this question, but I'll give it a shot. So normally you would use isocratic conditions when you don't have a very broad mix of compounds, or maybe only a few compounds, maybe like 5 or less. You could get away with using isocratic conditions, but in general, people typically use gradient conditions because they have a pretty broad range of polarities for their compounds, and they kind of want to speed things along, so that's why they'll use gradient conditions. So it's a little bit hard to answer the question unless you know how many compounds you're looking at and how close they are in terms of their hydrophobicity.
All right. This one says, thank you for your presentation. I'd like to hear any suggestions about how to transfer a method from an analytical to a semi-preparative column. How is the best way to employ this change?
Oh, sure. So one of the main things going from an analytical to semi-prep is the dwell volumes are going to be different. So my first suggestion would be to measure the dwell volume on the system where you're using the analytical column compared to the system where you're using the semi-prep column. So there are, method calculators where you can transfer the method. Just make sure you're using the same linear velocity on both columns, and that will give you a good chance at getting the same separation on the semi-prep column.
Okay, I think we're going to take one more question. I'm not totally sure what the question is asking, but it says to enhance resolution of the isomers, reduce pH.
I'm not exactly sure what the question is asking either, to be perfectly honest. Are they able to tell us what kind of isomers?
The full sentence is just, might have missed one of your last points. To enhance resolution of the isomers, reduce pH. We can go ahead and move on to a different question.
So my point there was really depending on whether the compound is ionizable or not is really how you should decide what pH to run at. So if you have acidic compounds, you would run at low pH to get more retention. But if you have basic compounds, then you would run at high pH to get more retention.
Okay, I think we'll go ahead and wrap it up there. Thank you, Dr. Schuster, for your fascinating presentation, and thank you, participants, for being a great audience. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. Thank you On24 for technology and production services, and thank you Advanced Materials Technology for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Gina Vitale. Goodbye.